Our next presenter is Sebastiaan Valkiers, presenting his tool ClusTCR.
Give me a second. Okay. Yeah. This one here. This one. Yeah. Okay. So, hi everyone. My name is Sebastiaan, and I'll be giving you a quick walkthrough of our TCR clustering algorithm, ClusTCR. So, ClusTCR, as you might expect from the name, is a TCR-specific clustering algorithm to cluster TCR sequences according to their putative epitope specificity. So we developed ClusTCR in order to address the huge complexity that we're facing with large TCR repertoires. And the way we made this so efficient is by dividing the clustering procedure into two steps. So in the first step, we effectively divide the large space of TCRs into smaller subspaces. We do this by generating numerical encodings for all the TCRs, and then using a very efficient k-means implementation from the FAISS library developed at Facebook to generate large superclusters, that's what we call them. So now we've essentially downscaled our large problem into smaller manageable subproblems. And for these smaller superclusters, we then construct similarity networks based on a Hamming distance equals 1 criterion. So we draw an edge between all the TCRs that are 1 at a distance away from each other. And then we apply graph clustering in order to resolve the final clusters. So this is how the algorithm works. And now I will walk you through some code examples of how you can actually use it. So if you want to follow along, just visit my GitHub, which is github.com/Valkiers, and navigate to the ClusTCR demo repository, which contains an IPython notebook with all the code examples I'll be showing here. So installation is very simple. ClusTCR is a Python package, and it can be installed using Conda. So just install it using the command right here, using the following Conda channels listed below. And for the sake of this demonstration, I'll be showing an example of a modestly sized TCR repertoire of about 100,000 sequences. So, like I said, ClusTCR is a Python package, so we can load it into Python. So we can import ClusTCR in Python, and specifically we will import the clustering class, which is the class we will use to perform the clustering. ClusTCR follows the scikit-learn practices. So for those of you that are familiar with that, this will look very familiar. So we can configure a clustering object and then fit our data to it using the fit function, similar as we would do in scikit-learn. So in this case, we use the CR3 sequences. This will generate a clustering result object, which we can then use to access our clustering results using the clusters.df property. We can also do some downstream analysis with this. We can, for example, compute numerical features, numerical representations that represent the clusters, or we can export the network to a CSV file using the export network function. So what happens under the hood is these two steps. So the first step uses the FAISS library to generate the superclusters. So these are essentially large clusters of TCR sequences. So we can configure this using the method, as you can see in the clustering object. And this is very fast, as you can see. Only took 2 seconds for 100K sequences. And then we can also do the second step individually. So by default, it uses the 2-step process, but we can configure these individually. So this is by specifying the MCL function. We can also go beyond the default settings to improve our clustering performance. For example, we can enable multiprocessing by specifying the number of CPUs. In this case, we use 8 CPUs to get about a 3.5 times speed improvement. We can also configure the size of the superclusters which is now using the FAISS cluster size parameter. We set it here to 3,000. By default, it's about 5,000. And this results in about a 4 times speed improvement. Lastly, something we've also been working on is including vgene information into the clustering. And this can be done by specifying the include vgene parameter in the fit function, and then specifying the CR3 and vgene columns. Small note on performance. So from our benchmarks, ClusTCR outperforms other clustering methods in terms of speed and scalability, while maintaining accurate clustering performance according to the current standards that we use. Now, finally, if you want to learn more about the downstream applications, how we can effectively interpret and apply the ClusTCR results, you can find me at the poster session tomorrow, so come talk to me at poster 203. If you don't want to talk to me, which is understandable, send me an email at this email address. So I'll be gladly accepting any questions right now. Thank you.
So any questions for Sebastiaan?
Hi there, thanks for the talk. Could you tell us a little bit more about how the superclusters are defined? What makes a supercluster a supercluster? Okay,
so like I said, I'll go back to this slide right here. So we start by encoding TCR, so the amino acids as numerical features. So we use the physical chemical properties to get a numerical representation, and then we use we get large vectors, and then we will use the k-means clustering algorithm to divide this space into smaller subspaces. So according to the size of the superclusters that you define, compute n clusters, which is just the size of your repertoire divided by the size of the superclusters that you define. So it will compute centroids, like k-means does, compute centroids, and then assign the vectors to these centroids. And that's, you basically get groups of TCRs that are roughly similar. But this way, since you get these groups of TCRs that are roughly similar, this makes it more manageable to work with, because you have smaller problems that you can solve instead of this one large problem.
So when you say that you can input in a particular CDR3 or a V, for instance, does that mean that that inputted V and or CDR3 is the center of a node, and then you're trying to use that as a reference point for the clusters that you want to build around that? Is that how that's working?
No, so essentially it just goes off of all of the sequences and doesn't really have it doesn't really use one sequence as a center point. K-means just initializes at a random point in the space, and then just converges until, well, it's an optimization process, so it converges until until all the clusters, all the sequences are assigned to a centroid according to the objective function.
One final question from the Zoom chat. How does adding the vgene affect the specificity of the clusters if compared to the ones without?
So including the vgene actually improves the clustering accuracy according to the current standards that we use. So we use the VDJDB to benchmark our algorithm, and it actually improves the clustering accuracy. Your clusters will be more specific. However, there's a trade-off, which we often observe. It's the cluster retention, as we call it. It's the number of clusters, the number of sequences that end up in any cluster that decreases when including the vgene.
That's great. Thanks so much, Sebastiaan.